Hello, good morning everyone. I'm Arkajyoti. I'm a graduate student of Biochemistry and Molecular Biology, and I am also working as a graduate research assistant in Department of Health and Biomedical Science. My thesis supervisor is Dr. Upal Roy. He is also assistant professor in Department of Health and Biomedical Science. My thesis, my thesis topic is biological optimization of antiviral of polymer-based antiviral drug towards the gut as with the lymphoid tissue GALT. So first of all, the HIV. The HIV or HIV-1. HIV means human immunodeficiency virus, which causes acquired immunodeficiency syndrome, which is known as AIDS, and it's transmitted in our body, though the biological fluid like semen, vaginal fluid, blood, etc. It also transmitted in our body by unprotected sex. It basically affected attack in our immune system. According to the WHO 2020, a report shows that 37.7 million people are living with HIV in the world, where 36 million people are adults. So it's alarming for the adult generation. If we presently there is a treatment procedure which is known as CRT, combination antiretroviral therapy, which is the combination of three or more antiviral drug, but this therapy has a limitation. First of all, this therapy do not kill the virus, only suppress the virus in the reservoir. And if any patient starts this therapy, he cannot stop the therapy. He should take the therapy until death. Because if you discontinue the therapy one time, then the virus rebound again and then affect our immune system. And we are trying to develop the nanomedicine using the nano carrier, because in comparison of traditional medicine, the nano carrier are flexible to carry hydrophilic, hydrophobic substance, both substance, and it's also have a high stability, high carrier capacity, also feasibility in various route like oral route, inhalation route, etc. And there are several types of reservoir in our body. The major reservoirs of HIV is brain, testis, dendritic cell, and GALT. But we selected GALT because in the several study, we see several study shows that a massive replication of HIV occurs in the mucosal lymphoid tissue like GALT. And in the GALT, we selected the M cell, which is the specialist cell of the mucosal tissue, which act as a gateway of the mucosal immune system, and it also sometimes plays an important role to transport the antigen through the small intestine to the mucosal immune system. In our study, we used the 6 formulation, which are divided into 2 groups. One is made of F127 polymer, and one is made of F127 and L621 polymer combination. And in 2 groups, we use 3 drugs. One is imitracetamin, which is annotated by FTC. One is tenofovir, which is annotated by TNF, and one is daltegliflozin, which is annotated by TLG. This formulation were prepared by our collaborator, Department of Molecular Pharmaceutics, University of North Carolina, Chapel Hill. So after getting this formulation, first of all, we do the we see the particle characteristics by PDI, polydispersity index, and the particle size. Then we see the cytotoxic activity of the formulation in different cell line. One is CACO2, which is human oncocell. The second is microglia cell, which is HMC3, and third is mouse macrophage cell, which is GSP raw, which is annotated by GFP raw. Even in the result section, we see that at the PDI polydispersity index of all the tested formulation within the standard range, which is 0 to 1. And that means it's follow the nano, all the formulation are within the follow the nano characteristics of the particle. And if we go through the particle size, amount of the formulation 1, 3, 5, and 7, and the particle range size range is 7 to 26 nanometer, which is within the range of the standard range is 1 to 100 nanometer. However, the formulation number 9 and formulation number 10 is relatively bigger, which is the range is 8 to 70 to 1200 nanometer, so little bit bigger. So if we go through the cytotoxic activity of our formulations, then we presented our result in the three category according to the cell line. So first of all, according to the CACO2 cell, the cytotoxic activity assay of the CACO2 cell of all the formulation, we see that the percentage of cell survival decrease in case of FTC in the highest concentration, which is 21 microliter. But in case of tenofovir, we see that the percentage of cell survival decrease from when we go through the lower concentration to higher concentration. And in the formulation number 5 is not no significant, but formulation number 7 is significant. But in case of GFP raw, the percentage of cell survival rate of formulation number 1, 3, 9, and 10 has no significance, but the formulation number 5 is significant. But in case of microglia cell, which is known as HMC3, all the formulation have no significant value. The no significant means they are non-toxic when they are used in they have accurate use. So it is safer for the cell line. So according to our above result, the chemical and biological characterization of our formulation reveals that they are the potent nanodrug delivery for the anti-HIV drug towards the guts. So our future plan is to do the in vitro cell uptake study, drug release study, and in vivo study on mice to establish our drug and maximize our potential use of the therapeutic agent for HIV patient. And I'm thankful to NIH for their grant, and also our also my lab mates and my supervisor Dr. Roy for helping me in my work and helping me in my work. Thank you to everyone.